Hello, everybody. I believe we are live, and I will see if that is the case in just a minute. Once it clicks through on YouTube over here, I'm just going to make sure that everything looks good before I officially get everything started. Yep, I see and hear myself. Great. Okay, so now I've muted myself, and we're good. So hello, everybody. Welcome to Creation Myths. So I'm Dan. I run the place, and I am here tonight with a guest. We have Otangelo Grasso here with us, and we are going to talk about irreducible complexity. So basically, what we're going to do is I'm going to take just a couple minutes to talk about how hypothesis testing works, like the general idea. I'm going to turn it over to Otangelo to talk about the concept of irreducible complexity. That'll take, I don't know exactly how long, 5 or 10 minutes. And then we're going to talk, I'm going to talk about irreducible complexity as a hypothesis. Basically, what we're going to do is examine this as a scientific hypothesis. Basically, what I want to do is strip away all the kind of baggage from this idea of irreducible complexity and just examine it on its own terms as a scientific hypothesis. So that's what we're going to do. So we have our brief little intro bits here, and then we'll just talk. And I anticipate we'll go until about 9 p.m., give or take. That's the idea. And yeah. So before I go any further, a couple quick announcements. First is that if you look down in the description of the video, I only updated it about 15 or 20 minutes ago, but there are a bunch of links that Otangelo has very helpfully provided on irreducible complexity, including one thing that is a specific response to me at some point. So if you go down there, there's going to be a lot of specific information. There's also his YouTube channel, and correct me if I'm wrong, Otangelo, I think your blog is in there. All your stuff is down in there, so everybody should check that out for additional, like, way more detailed information than you could fit in, a live conversation. So that's the first thing. Second thing is that before we get going, I just have a programming announcement that I'm really excited for, and I can't wait. On Wednesday, the 6th of January, I am speaking with the Dr. Ken Miller. That Ken Miller. We're going to talk about evolution and intelligent design and creationism and kind of that conversation, and kind of the approaches to that conversation and how they've changed over time. So that's what we're going to be talking about, and I'm really super excited to have that conversation. That being said, I'm also super excited to be having this conversation right here this evening. Let's get right to it. Like I said, I'm going to start with just a very short intro on the idea of hypothesis testing. So the idea here tonight is to look at the concept of irreducible complexity as a scientific hypothesis. And what you're trying to do when you come up with a proposed explanation or an idea or concept in science is you have this hypothesis, right? This proposed explanation, and we're going to test that. The way we're going to evaluate the validity of any hypothesis is to generate predictions that must be true, logically, if that hypothesis is valid, right? So what we're going to do... Oh, hang on.
Viu, todo mundo viu. Pois é.
There we go. Okay, so what we're going to do is... you're going to have your hypothesis, you make predictions, and then the idea is logically, you've probably all heard the thing with if-then statements, right? If X, then Y, right? That's your prediction. If whatever hypothesis is true, then blah, 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 right? And then you test the blah, 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 blah. That's your prediction, and you're going to see if that actually holds up, okay? So what you're trying to do when you're hypothesis testing is you're not trying to prove that your hypothesis is true. You're not trying to disprove competing hypotheses. What you're actually doing with hypothesis testing is attempting to disprove the thing that you have come up with. If my hypothesis is true, then X, Y, and Z must be observed, whatever the predictions are, right? And then if you don't see that, if you find things that contradict that, that invalidates your hypothesis. So what we want to do is follow that logical process, looking at irreducible complexity, right? So we want to examine this idea of irreducible complexity as a scientific hypothesis and examine how we would go about evaluating that. What predictions does it make? And then have those predictions been shown to be accurate or not? So some of you may be wondering, you've said this word irreducible complexity a bunch of times, but nobody has defined it yet. Can someone please define irreducible complexity? Yes, we can. For that, I'm going to turn it over to Otangelo. The floor is all yours. Take 5, 10, whatever you want to take, to tell everybody about the idea of irreducible complexity, and then we can bring this back around to looking at that hypothesis.
Okay, hello everyone. Good evening. Thank you, Dan, for having me first time at your channel. I hope that it will be a productive conversation, and eventually that we can have a follow-up. I'm not looking for confrontation or something like that. I'm looking just to have a nice conversation where we can figure out things. And if you have good points, I'm having no problem absolutely to acknowledge that and to give you a point. Okay, so you asked about defining irreducible complexity. I will here put how Behe defined it in the book Black Box, and then we can go from there. By irreducibly complex, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any of one of the parts causes the system to effectively cease functioning. An irreducibly complex system cannot be produced directly, that is, by continuously improving the initial function, which continues to work by the same mechanism by slight successive modifications of a precursor system, because any precursor to an irreducibly complex system that is missing a part is by definition non-functional. An irreducibly complex biological system, if there is such a thing, would be a powerful challenge to Darwinian evolution. Okay, so since you mentioned Ken Miller, which will be at your channel in a couple of days, I had actually an interaction with him a couple of years back at his Facebook page, and we had some back and forth, and then in the end, I gave as an example the way how Mycoplasma mobile moves, and to that he didn't give any answer anymore. So I would like to provide that example, in my understanding, as a prime example of irreducible complexity. So I'll share here the screen, and then I will present here the bacteria, and we can have a closer look on it. Okay, so I'll read here a syllogism and explain what it is about. Behe used as an illustration a mousetrap, where all the components are required for its function, and as such, it is irreducibly complex. Mycoplasma mobile is a bacteria which uses flexible legs that literally walk on the surface. Its biomotility mechanism is unique, and Mycoplasma mobile is propelled by GLI 345 protein legs, which repeatedly catch and release on the surface where they walk, and are driven by the force exerted by a so-called P42 protein through GLI 521 molecules, supported by surface structure on which they walk, based on the consumption of energy in the form of ATP. Each of the 4 proteins used for walking are essential. Mutation experiments in the last decade have demonstrated that these proteins each must be precisely and correctly sequenced, otherwise the function ceases and motility is not possible. 4 component proteins are directly involved in the mechanism, and at least 10 proteins are involved in the cytoskeleton. None of the 4 proteins has significant sequence similarities with proteins of any other bacteria, so there could not be co-option. And since none of these components can be dismissed without harming the motility function, this system is irreducible, interdependent, working as an integrated system, and on top, none of the components could be co-opted from other organisms. So here we have the illustration of this mechanism. There is one, the GLI349. That is the protein, the leg, responsible for binding on the surface during gliding and is required to convey movement. That is that red part here. Then secondly, the GLI521 is the motor, or the gear transmitting force from the motor to the leg, that green part here. Then 3, the GLI123, that part here, is necessary to stabilize GLI349 and GLI521 proteins. And P42, which is that part up here, has ATP's other activity. The energy for mobile gliding is supplied by ATP hydrolysis, and that the detachment from the solid surface is an energy-dependent process. So here is a small video where you guys can have a look how that works. Okay, so I would consider that as a prime example of irreducible complexity, where none of these 4 proteins can be removed, otherwise the function will cease to exist. The bacteria could not move anymore, and that is a system which, in my understanding, could not be the product of gradual evolution. Now, some remarks in regards of irreducible complexity, and then I will hand over to you then. Natural selection would not select for components of a complex system that would be useful only in the completion of that much larger system. In other words, why would natural selection select an intermediate biosynthesis product which has by its own no use for the organism, unless that product keeps going through all necessary steps up to the point to be ready to be assembled in a larger system? Never do we see blind, unguided processes leading to complex functional systems with integrated parts contributing to the overarching design goal. A minimal amount of instructional complex information is required for a gene to produce useful proteins. A minimal size of a protein is necessary for it to be functional. Thus, before a region of DNA contains the requisite information to make useful proteins, natural selection would not select for a positive trait and play no role in its guiding its evolution. Okay.
All right. Thank you for that overview. And especially the quote from Behe that you had at the beginning, defining irreducible complexity, is literally the exact quote I also have pulled up, because that is like the canonical definition from the 1996 book Darwin's Black Box, which I do recommend everybody read, despite disagreeing with it and thinking it is not very good. I do recommend everybody read to be familiar with Behe's argument. So I also actually want to start by saying I agree with that bit at the end there, saying natural selection with the implicit kind of on its own, can't do X, Y, and Z. And that actually brings me to kind of where I want to start, kind of talking about irreducible complexity. So the idea again is that this is a hypothesis, and Otangelo, tell me if this is fair. Basically, the hypothesis is that if you have a system that meets Behe's criteria for irreducible complexity, if it checks all those boxes, multiple interacting parts, they all are necessary. If you remove one, the system no longer works. The hypothesis is that if a system meets those criteria, then that system could not evolve. Is that basically fair?
Yeah, I have watched your video, which you have on your channel about irreducible complexity, and I have linked to my criticism. I have selected 3 points which I've addressed, and I would like to make just an observation in regards of this. You say literally this then: an irreducibly complex system cannot be produced directly, that is by continuously improving the initial function. Now, that is not what Behe actually said. He said that the initial function is what has to be explained. And in order for that initial function to be, it depends on several well-matched interacting parts. We see that all over in biology, where a specific purposeful function is only achieved when several well-matched parts interact with each other. And I think your criticism is quite interesting, where you say that the argument is like a guacamole. And I see it like that then, that while the majority of the intelligent design proponents have just selected a few examples of irreducible complexity, I have over the year actually collected many more. And I would say that irreducible complexity can be observed all over in biology, from biochemistry, from DNA to very complex systems like speech, which is an example which I brought up in my last debate at MDD. Why that? Because I mean, biological systems, there are all kinds of biological systems. And of course, most of them, they are not just single units of molecules, but they are integrated parts working in a joint venture together to have a specific outcome or a product. And
may I jump into, so I agree with you. That's the definition of irreducible complexity. So what I'd like to do is if we could really kind of try to tease apart this hypothesis, right? So we have this idea, you have this system, any systems, lots of systems that I think we would agree meet these criteria, right? For whatever function you want to talk about.
Sorry to interrupt you then. I have put here 5 singular points to which the argument can be subdivided. That is, first, a single system composed of several well-matched interacting parts.
So if I may, we're going to go through that. I can just kind of.
Okay.
So what basically the way there's 3 ways, as I see it, that we can interpret Behe's hypothesis as he wrote about it in Darwin's Black Box. Now he's very specific here, where he talks about well-matched interacting parts, basic function, removal of any one part. He talks about cannot be produced directly by continuously improving the initial function, which is really important. Basically, what he's saying is in the description that you provided, which is that's the original description of irreducible complexity. Behe is limiting his hypothesis to only talking about basic Darwinian mechanisms as Darwin would have understood them in like 1860. He's actually leaving out a lot of, I'm just starting a timer for myself so I don't just ramble. He's leaving out a lot of like other mechanisms of evolution. So he's leaving out things like neutral drift. He's leaving out things like recombination, gene duplications. Darwin didn't know any of that stuff. Those are non-Darwinian mechanisms. He's leaving out exaptation, where a system has one function and then it ends up doing a different function, right? He's leaving out things like neofunctionalization and subfunctionalization within proteins and protein families. So the first version of the hypothesis that if a system meets the criteria for irreducible complexity, it cannot evolve. The first version of that like kind of the strict interpretation of what Behe says,
there are four versions.
The question of like, could a thing evolve? Because it leaves out 150 years worth of subsequent post-Darwinian understanding of how evolution and evolutionary processes work. So that's the kind of the most, I think, kind of the most textualist interpretation, but I actually don't think that's the best way to interpret the hypothesis, because Behe was writing in 1996, not 1866. So obviously he's aware of like all these other mechanisms. So I think we have to kind of give Behe a little bit of credit and say, like, okay, we know the way you said it, it's kind of just specifically Darwinian, but we need to kind of, I think, reframe it a little bit, give Behe the benefit of the doubt here. What he means is that even if you include all these other mechanisms,
I think he thinks that.
Like, I think we can agree that that's kind of fair.
Unless he has explicitly outlined that he doesn't include the expanded synthesis and all these epigenetic factors and so forth, then of course that would be a point of criticism, which I agree with you. But I would say we have to include whatever is proposed as an unguided mechanism and go from there.
Great. So see if you agree with me on this then, because we're the idea here is we want to see how we could evaluate this hypothesis. So now we have two options. We had option number one. We've said that probably isn't right. So we have kind of what I call the strong version of the hypothesis and the weak version of the hypothesis. Now, the strong version of the hypothesis is that for any system that meets these criteria, it could not evolve through any evolutionary natural processes, right? And
I would not restrict it only then into evolutionary mechanisms, but unguided non-mental mechanisms in a very general sense. That includes also abiogenesis, which doesn't have evolution as a possible mechanism for because it requires DNA replication. But I would say we can even put here mechanisms that are not seen in biology and apply the same criteria.
Okay, so that's actually more expansive, and I'm down with that. Saying there's a whole bunch of processes, right? And you can include any set of those processes you like. A system that meets the criteria for irreducible complexity cannot evolve via any of those processes individually or in combinations. That fair?
Yes. The key issue is that no intelligence is involved.
Exactly. Okay, so that's the strong version. That's like the absolutist version of the hypothesis. Now that's a testable hypothesis. That's really nifty, right? Because now we're saying, here's the line: if you meet these criteria, it cannot evolve. Like, that's the strong version of the hypothesis. We're going to come back to that one because I think that's the important one. But the third kind of interpretation for this hypothesis is what I call the weak version, which it's all that same stuff. But it's the only difference is that you're saying if something meets that criteria, it may not be able to evolve. In other words, according to the version number 3, we're saying there are some systems that meet those criteria and cannot evolve, and there are other systems that meet those criteria and they can evolve, right? And now that version of the hypothesis,
I've never seen that kind of idea then. But a clear, and I would say there has to be made a clear drawing line. If the system is irreducibly complex, it cannot evolve. Right. If it is possible to evolve, then it is by definition not irreducibly complex.
So I want to put a pin in that because we're going to come back to that in a minute. But I just want to dispense with this third variant first, because you do see this not from Behe, but you do see it some places. And the problem with that third version is that if you could have a system that meets the criteria and it can evolve, and you have other systems that can't, that is not a falsifiable argument, right? It's just there's always going to be another system that we don't have a step-by-step explanation for its evolution yet. Therefore, we can say, well, that's the thing that prevents evolution from being able to universally explain biodiversity. So that third version, the weak version, is not falsifiable. So we can put that one aside as not a valid hypothesis. So I think we're in agreement that the relevant version of the irreducible complexity hypothesis is that strong version, the second version, that if it meets the criteria, can't evolve, period.
Yeah,
okay.
I agree with that.
So now I think the question is, let's test that. Right now what we want to do is be able to test that hypothesis. So the way to test that hypothesis is not identify things that cannot evolve, right? Because now what you're doing is trying to disprove the alternative, right? Trying to disprove evolution. But in hypothesis testing, what you're really trying to do is disprove the hypothesis itself. So the way to test the irreducible complexity hypothesis would then be to identify systems that meet the criteria for irreducible complexity and document their evolution. And if we can document their evolution in real time and observe it, then that's going to invalidate that hypothesis. I think
correct. Yes.
Okay, cool. So this is where we're going to hit our sticking point, because I think we have observed, like right now in real time, we're getting to watch systems that meet that criteria evolving. We're watching it happen right now.
Well, if you can show that systems are evolving, that doesn't falsify the hypothesis of irreducible complexity, because we know that evolution happens in nature. The question is, what kind of evolution are we talking about? If we are talking about wolves speciating to become dogs, then that is something that nobody has any dispute about. Okay, now if the question is of big changes, of things like primary speciation of macroevolution, where one organism gets legs and eyes and ears and very new complex systems from unicellular to multicellular, or photosynthesis or ATP synthase or the cell by itself. I can give you many examples. Then that is a thing that we can talk about, because it is disputed.
Well, so that's why I lean heavily on what we can directly observe, right? If we can, the important thing is to very firmly plant the goalposts and say, here are the criteria, which we've done. We've established the criteria. Now we can identify systems that meet those criteria. And as you said, there are tons of systems in biology that meet that criteria. There's a lot of stuff that meets those criteria. So the thing that we should be looking for is, have we observed in like human, since we've been looking, have we observed systems that meet those criteria evolving? And I think at many levels, the answer is yes. And I'll give just two examples very briefly. One is a molecular example. One is a little larger scale. On the molecular side, if people have seen me talk about various things before, you've maybe heard me mention this protein in HIV called VPU. It's a tiny little protein in HIV1 group M, specifically is the one I care about. This protein, it has one specific job in HIV and the SIVs, the simian immunodeficiency viruses that HIV1 group M evolved from. In HIV1 group M, it has a second function, it has two functions, and that second function is completely new. It inactivates part of the human immune system. It doesn't matter what it is, the specifics, it doesn't matter. It has a new function. And this new function actually requires that it forms a pentamer, a 5-unit ring in the plasma membrane of human, I forget what cells specifically, whatever cells HIV infects, whatever subclass of white blood cells. And so they've actually done the work to figure out exactly what mutations facilitated this new function. And it turns out there are two specific regions in this protein that allow it to accomplish this new function. There's three specific mutations in one of those regions that they've identified. Haven't identified the specific mutations in the other region, but there's a region, so it has to be at least one in the protein. So you have 4 specific mutations. And if you don't have all 4 of them, it doesn't form the pentamer, and it doesn't inactivate this part of the human immune system that's a little different from the chimp immune system. And we know that this new feature evolved, and it meets Behe's criteria. Well-matched parts, need to have all the parts for the function. There's no selection for the intermediate states. You need to have all these parts in order for it to work. And we know that this evolved within the last century because HIV only jumped into humans around 1930, give or take. And for this, every form of HIV, there's at least 3, possibly 4 distinct forms of HIV. HIV-1, that is. They all have ways around this problem. HIV-1 group M uses this VPU protein to do this specific thing. The other ones do it a little bit differently and not as well. And so we have this. We should be able to agree this is a molecular trait. It's irreducible. It has multiple components for this system. It has to interact with itself to form the pentamer. And then it has to interact with this other protein, this human protein, that the ancestral form of VPU couldn't interact with. So you have lots of mutations that are required in a multiple interaction system, but it all has to come together at once because they've done experiments on this. And if you knock out those mutations one at a time, it doesn't work. We can test that directly. So this is an irreducible system that evolved within the last century. We know it did. So like that right there should invalidate the hypothesis. That's we've witnessed and documented the evolution of an irreducibly complex molecular system.
Well, then I would not say that invalidates the proposition of irreducible complexity. It is just a system which can evolve. So it is not claimed that there aren't systems that cannot evolve. What we are saying is that are systems which are irreducibly complex, and there are systems which actually evolve. We are not claiming that evolution doesn't happen at all. Okay.
But does that system that I described meet the criteria for irreducible complexity? I think it does, according to these criteria that we agreed with earlier. I think that system, that VPU inactivating this protein called tetherin, that meets the criteria for irreducible complexity.
Well, if it is irreducibly complex, it cannot evolve by definition. But if it is evolving, then it is not irreducibly complex. You cannot have it both.
See, there's where you're moving now from definition 2 to definition 3, right? Because the point is that we established the criteria ahead of time. Now we're saying, well, because it evolved, even though it meets the criteria, it's no longer irreducible complexity. That's putting the cart before the horse.
Well, no, I disagree then, because an irreducibly complex system cannot evolve by definition. But at the moment that you can demonstrate that it has evolved, it is not irreducibly complex because it evolved. You cannot have it both ways.
I think what we're doing here is just, to be honest, I think we're moving the goalposts a little bit because we established the criteria and we agreed on the criteria, and now I'm giving you a system that we agree evolved that meets the criteria. But now let's move on from HIV. That's a small scale molecular system. It's not that many different mutations involved. What about something like like a placenta? Would the evolution of a placenta be irreducibly complex? Just to give everybody an idea, in order for placental gestation to work, right? You have your embryo, it has to implant in the uterine wall. The uterine wall has to be sufficiently full of blood vessels to actually provide enough nutrition for that embryo before it arrives. If the embryo arrives and that triggers we need to make blood vessels, too late, the embryo is going to die. Right. So the embryo you need to have the environment for the embryo when it arrives. Then you need to have mutual signals back and forth, triggering embryonic growth in a specific orientation relative to the uterine wall. You have to have growth of blood vessels in the uterine wall relative to the embryo. Eventually, you need to have a very dense capillary network that's intertwined, and just telling both sides of the equation to grow blood vessels isn't enough because it'll just be a tangled mess. They need to grow in a very specific pattern. You also need the embryo to suppress the maternal immune response, because what happens, for example, in marsupials, which are not, they have placentas, but they're not true placental mammals like eutherians. The uterus actually has a robust immune response to the implanting embryo, and it kicks it out very early. That's why if you Google like an image of newborn kangaroos, it's horrifying because they're so underdeveloped, and that's why they finish developing in the pouch. So you need to have some immunosuppression by the embryo acting on the mother, but not too much, because too much immunosuppression and now you risk killing the mom, right? You also need a system for regulating nutrient transfer back and forth, because the embryo would just take everything and kill the mom accidentally, right? So the mom has to have a way of limiting that. And if any one of these components of this system are not calibrated already, then you're not going to have successful development in utero. So I think placental development counts as an irreducibly complex system. Now, I'm sure everybody, and probably Otangelo, can guess the punchline here. What do you think it is that we are witnessing evolving in real time in lizard populations right now? What's everybody think? It's placental in utero development. It's true placentas in lizards. That's not something we're hypothesizing. It's not in the fossil record. It's something we're watching happen right now. That's the evolution of an irreducibly complex trait, a really complex morphological trait. And just like the HIV example, if we can document the evolution of, if we can witness the evolution of a trait that meets those criteria, then that invalidates the hypothesis. So that's my second fun example of like the actual observations that invalidate Behe's hypothesis.
Well, again, then, if it can evolve, it is not irreducibly complex. Then or you can say it is, in that case, it is falsified. Now, if there are cases where it is demonstrated that the system cannot evolve, then the hypothesis of irreducible complexity stands. And I brought forward exactly the example of Mycoplasma mobile, because it is a very basic system, just composed of 4 parts, which if you remove any of these 4 proteins, you lose the function. But each of these 4 parts, they do not have any other function in any other organism, and you cannot say, oh, that could have been co-opted from somewhere else. So in my understanding, that meets the criteria of irreducible complexity. Now, if you can demonstrate how that could have evolved, then of course that falsifies the example.
So can you explain why a placenta does not qualify as irreducibly complex?
No, because I have not studied what you just explained right now. So yeah, I would have to have a closer look to it and then give you an answer to it. But if it can evolve, as you said, in lizards, then it is not an irreducibly complex system. Then it can evolve.
So how specifically can I distinguish then between systems that kind of sort of look irreducibly complex but aren't, and systems that are truly irreducibly complex? Because
I just gave you an example.
Well,
you would have to try to demonstrate how the working mechanism of Mycoplasma mobile could be due to evolution.
With respect, no, that's not what has to be done, because that's flipping around how hypothesis testing works. The point it's not about, I have a hypothesis, now it's on everybody else to explain everything in the context of this hypothesis. It's, I have a hypothesis, but if we can find a contradiction for that hypothesis, then that hypothesis is invalid, right? That's how hypothesis testing works. So I don't know enough about that specific system in Mycoplasma to be able to tell you if we have a plausible evolutionary pathway to it. I don't know. I've never studied it. I can tell you for other systems that were claimed as irreducibly complex, things like the bacterial flagellum in E. coli, the blood clotting system in mammals, the immune system, any one of these various systems that, for instance, Michael Behe has claimed is irreducibly complex, we actually have a pretty robust evolutionary explanation for these. Now, I think in the examples I'm giving, I'm going a step further. I'm giving you systems that I think meet the criteria that we agreed on, right? And in particular, the VPU one is somewhat puzzling because it is, as far as we know right now, at least 4, possibly as many as I think there were up to 7 specific mutations that were identified, but there's at least 4 we know of for sure. And you just described a system that has, well, you need these 4 parts. Now, these 4 mutations in VPU facilitate 3 distinct new interactions, right? You can picture the VPU molecule, right? It's holding hands with one on this side, holding hands with one on that side, plus the pentamer has to interact with the human protein. So you've got 3 new interactions involving...
Well, then you are again explaining something which I am not even questioning. I'm not questioning your examples. And I think the best examples would be then those which there have been made experiments, knockout experiments, where, for example, one protein, which is part of the integrated system, has been removed, and then they have checked if the function has been preserved or not. And in regard of the working mechanism of Mycoplasma mobile, that has been done. They have removed each single of the proteins, and the function has been lost. And each of those proteins, they do not have any function by their own or in another system where they could have been co-opted. Totally. And I can give you many other examples if you want. And the best is that we maybe stick to things that you also are familiar with, so we can have a look on something that we both know. The 2 examples which you gave, I'm not familiar, so I cannot even object to those.
So I hear what you're saying, but I don't think going this example, that example, this example, I don't think that's fruitful. Because again, that's not how hypothesis testing works. It's not on, everybody else. If someone goes up in science and says, I got a new hypothesis, it's not on everybody else to conditionally accept that unless they can show that every question that hypothesis raises is answerable, right? That's not how hypothesis testing works. It's on the person stating the hypothesis to show that that hypothesis is consistent, makes accurate predictions, and makes predictions that are valid, that do not result in findings that contradict the hypothesis. So what I'm trying to get at here is that if we can identify systems that meet the criteria, which I think I've done twice now, and we could keep doing more. Another really good example.
No, I give you the 2 examples which you made. In special because I don't know them, so I cannot object them based on knowledge. So you can repeat them 3 or 4 times. I will not be able to object them.
I'm not going to repeat them. What about endosymbiosis?
But the Mycoplasma is in particular interesting because knockout experiments have been done, and it doesn't work without, if you remove any of the 4 proteins, it doesn't work. The working mechanism ceases to function. Then they have...
We've heard this already, so I'm agreeing with you. That system meets the criteria.
So I'll give you another one. And
Otangelo, my point is, going through the litany of examples is not relevant to the validity of the hypothesis. Let's look at something where we're familiar with, and we can, before we even talk about it, we can agree that it meets the criteria. And I think, what about endosymbiosis? A new membrane-bound organelle, right? Mm-hmm. Can we agree that that would meet the criteria for an irreducibly complex system? You have genes moving.
No.
Endosymbiosis into your genome.
No, I don't see how that would be irreducibly complex. I don't see that. But
I just want to be clear. Though, endosymbiosis one of the major evolutionary advances going from prokaryotes to eukaryotes, one of like the big things, that's not irreducibly complex. There's no barrier to that by irreducible complexity.
I think that hypothesis is doesn't work based on other reasons, which is not because of irreducible complexity. Okay.
Okay, so new organelles don't meet the criteria for irreducible complexity, even though you need to have specific proteins trafficked back and forth between the nucleus and the chloroplast, for example. You need to have selective genome reduction in the organelle. You need to have selective maintenance and getting rid of membrane components from that organelle. And if you mess up those membrane components, the organelle dies and it ceases to function. Like, we could just go on and on. There are dozens of things you need to have a functional organelle.
Well, the thing is, Dan, that eukaryotes, there are so many different kinds of eukaryotes that it is difficult to draw a line and say, okay, this kind of eukaryote is irreducibly complex, and then you come, oh, but it could have evolved like this and that and so forth. So I don't see that. I've never used that as an example, but I can give you a series of examples, and eventually, if you are familiar with one of the examples, then we have a common ground where we can have a closer look to it. And I can give you a few, and if you like it, then we can have a closer look.
This is the point, that this is my sticking point when it comes to irreducible complexity, is what you're describing is this game of whack-a-mole that I joke about. Presenting example after example of things that cannot evolve because they are irreducibly complex is not how you support the hypothesis of irreducible complexity. The way to do that is to examine what we have documented, like directly observed evolving. I think that's unnecessarily restrictive. I think we should be able to look at fossils, for example, and say like, oh, look, there was definitely a time when arms didn't exist, and then they did, therefore arms evolved, right? I think that's something we should be able to do. But for the sake of fairness to be, he, and to you, I'm saying, let's ignore that. Let's just talk about things that we have / are witnessing, right, in real time. And when you look at those things, what we can do is we can identify things that meet the criteria for irreducible complexity. And the way this hypothesis works that we agreed on is that if a system meets those criteria, right, that's the starting point. We're starting there. We don't know anything about its evolution. We're just saying if a system meets those criteria, right, then necessarily, according to the structure of this hypothesis, it cannot evolve. That's what we agreed on like 45 minutes ago.
Okay.
So now, if we can identify systems that meet those criteria, but instead of saying A, therefore B, it's A, therefore not B, that invalidates the hypothesis. If we can observe irreducible complexity.
Well, I can give, as I said, I can give you many examples.
Would you like examples where we don't have documented evolution? That doesn't matter one way or the other for the hypothesis.
Well. Then, if you have documented evolution, then by definition, that what is documented is not irreducibly complex.
Committing a very basic logical fallacy, and I forget the name of it because it's been so long since I took intro to logic. But so the hypothesis is if A, then B, right? And what we can say is if not B, therefore not A, okay? Or therefore A is wrong, right? If not B, then, if not, I forget the logic of it. It's if not B, therefore not A, right? Because
I would like to make, then let me give you an example.
Irreducibly complex.
Then let me give you an example.
I think systems that meet the criteria, it doesn't matter for the hypothesis testing. That's the point.
Okay.
We should be able to agree on certain systems that meet the criteria, right? So like, I think that VPU protein in HIV meets the criteria of irreducible complexity with its new function.
First of all, nobody is claiming that evolution occurs in biology. But what is being claimed is that there are systems which could not have been the result of evolutionary processes, and we can test that by knockout experiments. I gave you an example with mycoplasma. I gave that example to Ken Miller. He did not even try to reply to me and say why that system can evolve. But I would like to give you another example to just to make clear what we are talking about here.
In the chat, by the way, everybody, if you want to see it, CRISPR's got me covered. Thanks, CRISPR. What's the fallacy there? There's a fallacy related to that. I forget what it's called. Sorry, go ahead.
So let me suppose that I want to make an analogy here. Let me suppose that you want to make a project for a photovoltaic system, okay? And you need 5 parts which are working together in order to store and have energy available to that system. And the first part is the solar panels, okay? So you have the solar panel, then you have another 4 parts of the system, and at the end you have the batteries where you store the energy of solar energy. Then I would like to make a point, and if you don't let me talk, you will not follow what I am saying. When I finish, then you can criticize it. Is that okay?
Absolutely. I'm already agreeing that whatever you're describing is irreducible. I'm on board.
Okay, so let me finish, and then you can criticize it if you do not agree with what I am saying. Okay, so you have 5 parts, and the first part is the solar panels. But in order to have the function, the minimal function, which is to store energy usable for your house, for example, you need 5 parts. The photovoltaic system needs 5 parts. The solar panels, they require a complex factory to make these solar parts, these solar panels. Okay. Now, do you think a designer would design a complex factory to make solar panels which have by themselves no use unless you have all the other 4 or 5 parts in order to have at the end energy disposal for whatever you use that energy for? Do you think it would make sense to make solar panels which by themselves have no function?
Oh, absolutely not. That wouldn't make any sense at all. I agree. It's an irreducible complex system. 100%.
Okay, great. So this is perfectly an analogy to what we see in photosynthesis. And in photosynthesis, the function of solar panels is performed by chlorophyll A. So what happens? Chlorophyll A, in order to have function, it must be embedded in the light harvesting complex, and you need about 25 different protein complexes to have in the end glucose. Now, in order to have chlorophyll B, you need 17 complex proteins, and of those, the 7 last proteins, they are exclusively used only in that biosynthesis pathway to make chlorophyll. You cannot use these proteins. They are not used in any other system in biology. They're exclusively used in the biosynthesis of chlorophyll. So I would say that this is an irreducibly complex system, because the chlorophyll biosynthesis pathway, if chlorophyll by its own has no function, the whole biosynthesis pathway would have no function either. So why would evolution produce such a complex system if there would be only chlorophylls but not all the rest of the photosynthesis pathway?
Yeah, that meets Behe's criteria of irreducible complexity. I agree, absolutely. So let me put this to you a different way, though. And then what I'm gonna say this, and then I'll give you a few minutes to wrap up because we're coming up on 9 p.m. here. So I'll give you a few minutes to kind of make your final point after this if you want.
Okay.
As much time as you want. So what you're describing here is basically the third hypothesis, the third version of the hypothesis that we discussed at the top, rather than the second. So we've kind of shifted from the strong version of the hypothesis that no systems that meet Behe's criteria can evolve, to the weak version of the hypothesis, that there are some systems that meet those criteria that can evolve and some systems that cannot. For example,
I'm getting your point now about weak and strong and so forth. Okay.
So if I may, so like, for example, that HIV protein I told you about, when you describe with the mycoplasma, you can do knockout experiments and show that you need each piece. It's an irreducible system. You can do the same thing, and they have done the experiments for this with that little VPU protein in HIV group, HIV type 1 group M, and shown that you need each of those mutations to have this new function. It's exactly the same as the knockout experiments you've talked about with mycoplasma to establish that trait as irreducible. We've done this with that HIV protein and shown that it is in fact an irreducible trait. Now, let me just kind of slightly kind of take what you just said and just kind of turn it around just a little bit, because you just described in some detail photosynthesis. Like, okay, what happens if you have a chlorophyll without the associated electron transport chain, or the electron acceptors, the electron donors, right? You need a way to get the electrons. Why would you have a way to get electrons out of, for example, water without the rest of the system already there, right? It doesn't, but the system doesn't work without being able to get those electrons in the first place. It's irreducible. Okay, but let's talk about something like a placenta with that. What's the point of having a uterus that grows very thick and has lots of blood vessels if you don't have an embryo that knows to implant in the walls of that uterus? What's the point of having an embryo that's capable of that if you don't have an organ that can accept it and assist it in growing? What would be the purpose of an embryo that can signal to the mother to, hey, tone down your immune response so I can camp out here for a little bit, if it's not going to implant in the wall of the uterus anyway? And we could just keep going through those structures, and you could do it for endosymbiosis as well. What's the point of having an organelle inside the cell, a larger cell, long term, if you don't have an interface between those two structures, the nucleus and the organelle, right? You could go through all of these different systems.
Yeah, for me, endosymbiosis is a falsified hypothesis.
The challenge for Behe is that we're observing all these things happen. We're observing in real time lizards evolving placentas. We know when HIV evolved, so we know when that new trait appeared in VPU. We are right now watching like a number of animals, like not just one or two, but like a half dozen or a dozen different species of animals being in different stages of acquiring photosynthetic organelles. Like, there are some animals that have genes for photosynthesis in their nuclear genome. There are other animals that vertically transmit the organelles from parent to offspring. And you could look at the eggs, and the eggs are green because they have the organelles. So not just stealing them from their food, they're actually vertically transmitting them. So we have these irreducible traits, but we're watching them happen. We're watching them evolve in real time. And if the hypothesis is that it's that strong version, right, the second version of the hypothesis, nothing that meets these criteria can possibly evolve because it's irreducibly complex. If that's the version of the hypothesis that we're going with, then that hypothesis is falsified because we're observing those.
Yes, I agree with you. I agree with you then.
Which means, and this is the last thing, and then I'll turn it over to you and you could wrap up with whatever you want, which means that what the argument that I think you're actually making is the third version of the hypothesis. That there are,
I'm getting you, and I agree with you.
Okay.
Yes.
So that there are some traits that meet the criteria that can evolve, but there are other traits that meet the criteria that can't. Now, the problem there is that that is an unfalsifiable hypothesis.
No, I disagree with that. I disagree with that.
The reason it's and again, I'll let you take as much time as you want, but the reason it's unfalsifiable is that you can always point to another system we have not explained and say, but this thing cannot evolve. And then when we figure out how that evolves, oh, but that other thing cannot evolve. Oh, but there's always going to be something else to point to. But for the hypothesis to work, and I'm going to read specifically what Behe says. It's the last thing you said when you quoted Behe, right? He said, an irreducibly complex biological system, if there is such a thing, would be a powerful challenge to Darwinian evolution.
Yeah, and I agree with that.
To be true, it must be the case that these types of systems cannot evolve, period. If some of them can and some of them can't, then then it's not a barrier to evolution, because we accept that some of those systems can evolve. You just follow the evolutionary pathways through the ones that can evolve.
Well, the question is, what kind of evolution then are we talking about? And I think that if we are taking, for example, photosynthesis, you know that photosynthesis is very important for advanced life.
One more minute, and then I'll give you as much time as you want to wrap up. So the thing where I want to land with that is, once we get into that third version of the hypothesis, we're no longer dealing with something that conforms to how science works, right? We're no longer dealing with a hypothesis that we can go into a lab and say, this is a specific prediction that must be accurate if this hypothesis is true. Because we could find things that actually violate the hypothesis, but don't, in theory, invalidate the hypothesis. And in order for a hypothesis to be a valid scientific hypothesis, the way hypothesis testing works, you need to have predictions that necessarily are true if that hypothesis is valid. And if we're moving from the second version of the irreducible complexity hypothesis to the third, then it no longer follows those rules. And that puts it, you can make the argument, but it becomes a philosophical argument, not a hard science hypothesis that we can actually evaluate experimentally. And for that reason, that's like the big thing. Version 2 is false. It's been falsified. Version 3 is unfalsifiable. And for that reason, it's not a valid scientific hypothesis. So with that, I'll let you take as much time as you want to kind of get in your
I disagree with you, Dan, because, for example, photosynthesis is absolutely essential for all advanced life forms. And science doesn't have any explanation whatsoever. Blankenship is the worldwide probably best expert on photosynthesis. He has written books on that subject. And photosynthesis meets that criteria, and you say it cannot be falsified, but it has been tested. And indeed, for example, the oxygen evolving complex, which splits water and produces oxygen, which is a key function of oxygenic photosynthesis, it has 3 proteins, which knockout experiments have demonstrated that all of these proteins, they are essential for the function. And also the cofactor, the metal cofactor, which is also involved in water splitting, is also essential. And these parts, they could not have been the product of evolution, because they also could not even have been co-opted, because they are not used in any other system. So they cannot be a product of evolution. And if just this is demonstrated, then in an overall sense, you cannot say that non-mental or non-intelligent evolutionary mechanisms explain biodiversity. You cannot say that. And there are other examples, like the origin of life, which is also requires several parts which are by themselves have no function, but they work only in a joint venture with all the other parts, and they could not be the product of natural mechanisms. And I think this falsifies the claim, which is basically the major claim, that no intelligence is required to explain the natural world and life and biodiversity.
Okay. Well, thank you for that summary. So everybody, that is where we're going to leave it. Otangelo, thank you so much for coming on. I hope that was enjoyable. I thought that was enjoyable. I hope everybody found that conversation entertaining and hopefully informative and maybe enlightening if we're really lucky. I want to remind everybody that in the description down below, we've got a bunch of links to Otangelo's various materials, so stuff he's written, his YouTube channel, it's all down in the description. So if you want more details on anything that he has talked about tonight, obviously we can only get into so much like actual biochemical detail in an hour-ish long conversation, but you could do a lot more in writing. So that is down below. And then the last reminder I will give you is on Wednesday, 1 p.m. right here, Dr. Ken Miller. We're going to be talking about evolution and intelligent design and creationism, and it's going to be just as fun as this was tonight. So thank you all, everybody. If you don't already subscribe, please subscribe. If you haven't liked the video, like the video, because this was a fun conversation. And that's it, everybody. So I am going to sign off. Have a great night, everyone. Now I just have to find the end broadcast button and click it. Good night.